Today we will study lecture 4, BBT315 Metabolism. Okay. In this lecture we will study catalyst. So let's say, what is catalyst? The catalyst are the chemical compound that accelerate the rate of biological reactions. So to understand the nature of catalyst, we can give a simple example, like think of sugar, and our bodies use it to give energy within a second and converting it carbon dioxide and water. But a bag of sugar can be on the shelf for years, even though the process is very thermodynamically favorable. The reaction will not break down to carbon dioxide and water. So what are the differences? The difference is catalyst. Okay. So the biocatalyst, also known as enzymes, here all living organisms must be able to self-replicate and catalyze chemical reactions efficiently and selectively. So enzymes, from Greek word enzymos, are the chemical catalyst of biological system. Enzymes have extraordinary catalytic power, often far greater than that of a synthetic or inorganic catalyst. They have a high degree of specificity for their substrate, and they accelerate the chemical reaction rate tremendously. And they function in aqueous solution under very mild conditions of temperature and pH, and unlike many catalysts used in organic chemistry. In many diseases, the activity of one or more enzymes is abnormal, and many drugs act via binding to enzyme. So in case, let's say, lactose intolerance is a disease where the lactase is abnormal. So this is a disease due to the abnormality in the enzymes. Okay. So what are the chemical features of enzymes? Enzymes are central to every biochemical processes. Acting in organized sequences, they catalyze the hundred of stepwise reactions that degrade nutrient molecules, conserve and transform chemical energy, and make biological macromolecules from simple precursors. Mostly all enzymes are protein except ribozymes, and their catalytic activity depends on the integrity of their native protein conformation. As you know, enzymes are protein. And if an enzyme is denatured or dissociated into its subunit, catalytic activity is usually lost. If an enzyme is broken down into its component amino acid, its catalytic activity is always destroyed. Thus, the primary, secondary, tertiary, and quaternary structures of protein enzymes are essential to their catalytic activity. Some enzymes require no chemical groups for activity other than their amino acid residue, but others require an additional chemical compound called cofactor. Those are either one or more inorganic ions like iron, magnesium, manganese, zinc, or a complex organic molecules called coenzyme. Coenzymes act as transient carrier of specific functional groups. Most are derived from vitamins, organic nutrients required in a small amount in their diet. Some enzymes require both a coenzyme and one or more metal ions for activity. A completely catalytically active enzyme together with its bound enzymes or metal ions is called holoenzyme. The protein part of the enzyme is called apoenzyme or apoprotein. Finally, some enzymes proteins are modified covalently by phosphorylation, glycosylation, and other processes. Many of these alterations are involved in their regulation of enzymatic activity. So here is the image of holoenzyme, and this is the cofactor, this is the coenzyme, and this is the apoenzyme. That means in holoenzyme, The protein part is called apoenzyme. Here are some inorganic ions that serve as a cofactor for enzymes. Like you can remember the first three, copper, which is a cofactor for cytochrome oxidase, iron cofactor for cytochrome oxidase or catalase or peroxidase, and very importantly potassium for pyruvate kinase. Here are some coenzymes that serve as a transient carrier of specific atoms or functional group. Let's study the enzyme classification. Many enzymes have been named by adding the suffix ase to the name of their substrate or to a word or phrase describing their activity. So biochemists by international agreement have adopted a system for naming and classifying enzymes based on the type of reaction they catalyzed. Each enzyme is assigned a 4-part classification number and a systematic name which identifies the reaction it catalyzed. Let's say here glucose react with ATP produce glucose 6-phosphate. The formal name of the enzyme is ATP glucose phosphatase phosphotransferase, ATP glucose phosphotransferase, and common name is hexokinase. And the EC number, that means enzyme commission number, is 27112. 2 means the transferases, 7 means phosphotransferases, 1 means phosphotransferases with hydroxyl as an acceptor, and 1 means D-glucose is the phosphoryl group acceptor here. So here is the international classification of enzyme. Let's say oxidoreductase. So you can remember this using a term oxidation and reduction, where hydrogen plays a very important role. That means oxidoreductase, those enzymes transfer the electrons or hydrogens or atoms. Transferase, those enzymes transfer groups or group transfer reactions. And hydrolases, those enzymes catalyze hydrolysis reactions. That means they lyse something by transferring water to the functional group. Or liases, they cleave carbon oxygen carbon nitrogen bond, and isomerase, they transfer the group with molecules to isomeric forms, and ligases, opposite to liase, they form carbon sulfur carbon oxygen carbon nitrogen bonds. And why biocatalyst over inorganic catalyst? So they have greater reaction specificity, they avoid side products, and milder reaction conditions conducive to conditions in cell, and higher reaction rate in a biologically useful time frame, and capacity for regulation control of biological pathways. So enzyme makes the desired one most favorable. So this is the end of the class 4. Let's see in the class 5.